Hi, you cool cats and kittens. It's Professor Osmundson, Joe the Science Ho, here from my home classroom. Students, kids, I hate this. I've been teaching here for, 6 weeks? Something awful. I'm teaching a class this semester on molecular biology, right? And we do this, sort of 2-lecture thing in molecular biology every semester where we talk about a thing called post-transcriptional gene regulation. This is how a cell is actually able to, even after it decides to turn a gene on, maybe decide it's going to turn that gene off. And we're in the middle of a COVID crisis right now, and I decided instead of talking about how cells do that typically, let's spend these 2 classes talking about how viruses manipulate cells, right? So how viruses actually are able to manage to do crazy things to the cell's machinery. And I decided, why not talk about what we know about coronaviruses, right? Coronaviruses are obviously having a huge impact on our daily lives. And I wanted to share with my students that the tools of molecular biology that they've been learning about all semester are the exact tools that we're using right now to try to understand how this virus functions. If you understand how the virus functions, you can better build sort of medicines to treat it. So, I did this for my students, and I wanted to share. I know that there are a lot of people, whenever I talk about this virus online, a lot of people get super interested. So I said to myself, self, why not share this with the internet? Why not, give everyone out there with kind of enough biology tools that they understand some things about what the DNA is, what an mRNA is, and how a protein product is made. The information about how this virus breaks all of the rules that they think they know about biology. Right? So that's what we're here to do today. We're going to talk about some viruses, right? Molecular biology of viruses. We're going to talk about how viruses, get into the cell, but then mostly we're going to talk about specifically how coronaviruses are able to actually get their genes expressed, get into the cell, get their genes expressed, right, in a couple of waves of gene expression, take over the machinery of the cell, right, and then leave the cell, leaving the cell behind to die, little bastards. So I wanted to start with coronaviruses by sort of showing where they live in this whole crazy world of viruses. So viruses, they're really interesting. I've studied viruses since I was an undergrad, actually, and I am almost 40. Right? In the whole big wide world of viruses, they do cells have rules, right? Cells encode their genetic material in DNA. Basic rule of life. No living organism that we know of does anything else other than encode their genetic material in DNA. That's it, right? Cells turn on genes by making RNA. That's what RNA does, right? It has some other rules, but generally, genes get turned on when RNA is made. Viruses go, hell no. I don't want to do that, right? Viruses have all of these mechanisms that do not follow the rules of other living organisms. Well, viruses are not living organisms, but another story for another day. So, if you dream it, a virus can do it, right? If you can imagine a genome made of RNA instead of DNA, a ton of viruses like influenza, right? Hepatitis, right? Tons and tons of viruses have RNA genomes instead of DNA genomes. HIV, right? Retroviruses, RNA genomes. There are also, DNA viruses. So there are single-stranded DNA viruses and double-stranded DNA. There's every type of thing that you can possibly imagine. Herpes viruses, right? Herpes viruses are double-stranded DNA viruses. They actually get into your cell, get into your genome, and stay there for the rest of your life, right? Influenza and coronaviruses are what we call single-stranded RNA viruses, okay? These are viruses who encode their genetic material in one strand of RNA. And how a virus encodes its genetic material is incredibly important for what we call the viral life cycle. How the virus gets into the cell and how the virus gets back out of the cell. I always ask my students questions in the middle of a lecture. I say, hey, students. And then students go, hey, Professor Osmundson. I'm laughing at all my awesome jokes. I get crickets here in my house. I hate it. Right? I'm gonna ask you a question. If you are an actual molecular biology student, pause the video, answer the question, think about it a bit, right? So I asked my students, I said, hey, students, right? If you can think about the life cycle of a single strand of RNA virus, if I have a strand of RNA, what does it have to do once it gets in the cell, right? Does it have to copy itself, right? Does it have to make RNA molecules that become a protein? Can it become a protein itself, right? If you have a DNA virus, what does it have to do? Does it have to make RNA before it can do anything else? Can a DNA virus make proteins directly from itself? Right? So just let it massage in your brain, massage in the brain, right? What does a single-stranded RNA virus have to do once it gets into the cell? Pause your video. Answer the question. Let's think this through together. I'm an RNA virus. I'm single-stranded. The cell uses RNA to make proteins. Theoretically, I could get into the cell as my single-stranded RNA virus and immediately, with no other machinery at all, go ahead and start turning myself into proteins, right? Those proteins, it might be smart to make ones that can actually then copy myself, right? So a double-stranded DNA virus is not going to be able to do this, right? A double-stranded DNA virus is going to have to get in the cell and get RNA made that will get protein made, right? So that it can actually copy itself, right? So different viruses, different strategies. For RNA viruses, coronaviruses are massive. They are big. They're about 30,000 letters, right? As a comparison, HIV is around 10,000, and flu is just slightly larger than that. So here we're talking about an RNA virus that, for RNA viruses, has a big, big genome. And what we mean when we say genome is the number of letters in its genetic code. 30,000 is huge for a virus. You out there? My students will be laughing hysterically at my awesome joke. You out there have 3.2 billion. 3.2 billion letters in your genome, right? So the recipe for you special snowflake, the recipe for you is 3.2 billion. And yet this virus, HIV, fewer than 10,000, flu around 13,000, coronavirus is around 30,000 letters. That's all it takes to copy itself, right? And somehow, one of the reasons I've been fascinated with viruses is that they're small. They are small. It doesn't take much to encode for themselves, and yet they somehow can do all of the things that are necessary to get into your cell, copy themselves, get out, and that can have huge ramifications for your health, right? The virus can kill you. Its little 30,000 letters are incredibly dangerous, okay? So again, here on this slide, I'm saying DNA, mRNA, protein, right? That is what we call the central dogma of biology, right? Genes are encoded in DNA, right? That DNA, when you want activated gene, becomes mRNA, right? And that mRNA is turned into protein, and that protein is an effector molecule. It goes and does stuff, right? It moves or holds the cell up or copies things, right? And, cells more or less play by these rules. And viruses in general, and the coronavirus in particular, LOL, nope. The virus goes, I don't want to do that, right? Viruses evolve mechanisms to do, all sorts of things with their genetic information. Right? So viruses do not follow the typical rules of cell biology and the molecular biology of the cell. That's why studying their molecular biology, number one, is really cool because they do so many things. And number two is really frustrating because every virus will have a different way sort of to subvert these rules, right? Every virus will have evolved a set of solutions to the problem. And just because one family of viruses, like the coronaviruses, have evolved these sets of mechanisms, influenza, which is another single-stranded RNA virus, will do it differently. Okay, so you really have to study each particular virus and its molecular biology in great detail. So here, this is like a classic virology slide, right? This next slide. What is the life cycle of this virus? How do its molecules get in the cell? Once it's in the cell, how does it copy itself to get back out? Okay, so let me get my pen back, my power on my iPad, which you're looking at, I think, is it over there? Is that over there? I don't know, right? The virus attaches to the cell through a specific interaction with its receptor. One of the cool things about this coronavirus crisis is that within just a few weeks of understanding that there was this new virus being transmitted amongst people in China, right? Scientists had figured out that it was a SARS-CoV virus, so a coronavirus, right? They had actually sequenced its entire genome to understand where it likely came from. And not only that, right? In this first paper about the genome, so the sequence of letters that this virus is made up of, they developed tests both for its genes and for antibodies that the body uses to respond. And they determined, right, what receptor it uses to get into the cell. They had a pretty good idea because the receptor for SARS-CoV-1, so the original SARS virus, the receptor was known. It's a protein called ACE2, right? And so they tested, does SARS-CoV-2, does this new SARS virus use the same receptor to get in the cell? And the answer is yes. Yes, it does use the ACE2 receptor to get into the cell. So it uses the ACE2 receptor to get into the cell. And that's, really a field we call cell biology, sort of how molecules traffic around the cell. And I'm not going to focus on that, right? So it gets into the cell through a vesicle and then has to get its RNA out into the cytoplasm, the sort of soup of the cell. I'm not going to talk about that. That's other scientists' sort of field of specialty. I'm going to talk about what happens to the RNA once it gets out into the cytoplasm. All right. Clear, pen, markings. Right? So the RNA gets out into the cytoplasm. Because it's a single-stranded RNA, it can act just like an mRNA in the cell. And again, my good students, crickets, crickets, crickets. My good students, what does an mRNA need in a eukaryotic cell to be translated? It needs a moiety on its 5' end and a moiety on its 3' end. Pause your video and tell me what those are. Okay, Professor Osmundson is very serious. Right? On the 5' end, we need a cap. And on the 3' end, we need a poly A tail. Well, guess what? The genome of a coronavirus has a cap on its 5' end and has a poly A tail on its 3' end. It, as soon as it gets in the cell, is a substrate for translation, right? So going from RNA to protein. And that is the very first thing that it has to do. So it doesn't have to encode anything else in its virion. That's what we mean when we say virion. That means like a viral particle. It doesn't have to have any other protein in there. It itself is a sort of molecule that the cell can recognize. The cell's ribosomes can recognize and immediately start translating and making proteins, right? Can we talk about my hair? I meant to do this whole thing with a hat on. Now, but I can't put it on in the middle. My hair is a disaster. Professor Osmundson doesn't normally look so disheveled. Okay, she's back. We talked about how once the RNA gets in the cell, right, it can immediately start making proteins from itself. And if we look at the map of the genome of SARS-CoV-2, which is the same as the map of the genome of SARS-CoV-1, right, we can see that there's a couple of big genes, massive genes, right at the 5' end of the genome, okay? And then there's a bunch of other genes at the 3' end of the genome. We'll talk about those later, all right? And the SARS-CoV-2 genome is sort of organized in this way, where it has at its 5' ends, okay, proteins that it needs to copy itself. And those are going to be made, bam, right, as soon as it gets into the cell, right? It gets into the cell and says, man, what do I need to copy? I need to copy myself. And it makes these proteins, all the proteins required to sort of copy and make its other genes, all right? And what gets made later are all of the proteins that it needs to get out of the cell, right? So it has these sort of two waves of gene expression, right? And even before this, I just want to talk briefly, just because there's some like really cool science that was done about, again, sort of how the virus gets into the cell, which from a molecular biologist point of view, we think of as like step 0, right? It's before the molecular biology happens. It's viral attachment to the cell, right? And so this, again, is recognizing the ACE2 protein, and it's done through a protein on the virus called the spike protein. And again, like, man, so quickly after we did the discovery of this virus and the sequencing of its genome, so that will tell us sort of what amino acids, what the protein is made of for the spike protein. Then a structure was solved of what it looks like using a technique called EM, right? So even the structural determinants of how the virus gets into the cell, we're starting to understand pretty well. All right, so RNA gets in the cell. Immediately a bunch of proteins can go ahead and get made, right? Those proteins that are made then have the job, get my pencil back, I'm so bad at technology. All right, of making the replicase. The replicase is the machinery, you've probably heard about this, the RNA-dependent RNA polymerase. RNA-dependent means it's using the viral RNA as its template, and RNA polymerase means that it's an enzyme that makes more RNA. Now again, different viruses do different things. A lot of viruses want to go into the cell's nucleus because the cell's nucleus has a lot of the machinery that the cell uses to copy its own DNA and to make RNA and all these things. So there's a bunch of, sort of machines in the nucleus that the cell has that the virus can borrow or co-opt, right? SARS-CoV-2 does not do this. SARS-CoV-2 has its entire life cycle in the cytoplasm. This is actually crucially important. Everything that the virus needs to do, that the cell would do in its nucleus, it has to do for itself, right? What does the mRNA need again? On its 5' end? Students. A cap. On its 3' end? A tail. The virus has to do those for itself because the cell will be doing those to the cell's RNAs in the nucleus, right? So, this again, it presents a particular set of issues, of problems that are unique to this type of virus. All right. Blammo, right? What does the viral genome need? It doesn't need anything. It itself is an mRNA, okay? Then the first proteins that are made are two massive proteins. This is ORF1A and ORF1B. Blammo. Again, cell has a rule. One mRNA, one protein, right? Eukaryotic cells, not polycistronic. One mRNA becomes one protein. You want to make more than one version of that protein, right? You got to make more than one mRNA. Cellular rule. Virus says, I don't think so. I'm not going to do it that way. The virus has one very long RNA molecule, that's its genome, and the ribosome gets on, and it starts going, and it's making a protein. Students are laughing hysterically. Dave, put in hysterical student laughter. Please, Dave. Right? And then it has this structure in the middle of its RNA. My ribosome is chugging along, doing its ribosome thing. It hits this sequence in the middle of the mRNA, right? And the sequence has a little thing that makes the ribosome kind of slip a little bit, followed by a crazy structured RNA. Remember that because RNA is single-stranded, it can base pair to itself. And it'll do so, but typically it'll do so and then fall off, then do so and then fall off, right? But in this case, it makes an RNA structure that is very, very stable. Okay? That RNA structure that's very, very stable messes up the ribosome. So the ribosome is moving along, and it's doing its ribosome thing, it's making a protein, right? And it slips, and then it hits this structure, and it goes boop. Okay? It just moves back by one base. And as all of you good molecular biology students out there know, right? RNA is, our RNA its information is encoded in codons that are 3 bases long, right? So if I change by one base, I actually shift the reading frame from reading a particular set of codons to whoop, one base back. And now I'm reading a whole new set of codons. This is called a -1 frame shift. This is the way that the viral RNA, this long 30 kb molecule, even in this kind of big 5' end, encodes 2 proteins in a way that's polycistronic, right? Again, cells don't do this, right? This is breaking the rules. And viruses do crazy things like this to translation, right? Where it manages to sort of get its stuff made. Okay. So, 2 polypeptides from a single mRNA. What crazy nonsense, right? It is truly, cells don't do it. Breaks the rules. All right. I believe in data. I'm a scientist, right? So, I told you that, but I want to show you how we know that's true. First of all, we have a structure. Get my pen back. We have a structure of the RNA, right? So we actually know what this crazy triple helix. How many helices are there normally in a helix? There are 2. The DNA double helix. Famous AF, right? Here we have actually a triple helix structure. So the RNA is folded back on itself in this really tight loop. All right? And that's given by this sequence that we talked about in the last time. And of course, if a scientist wants to show that a particular sequence is necessary and sufficient, well, it's going to mutate that sequence, right? And move that sequence around. So here's an experiment where this sequence, the slippery sequence, was mutated to all sorts of different other things. And this is an SDS page gel of in vitro translation. So it's looking at the protein product, right, of the ribosome. And you can see here there's one band, right? If I mutate that slippery sequence, the ribosome just keeps trucking along and it hits a stop codon, falls off, right? But if I keep in that slippery sequence, like over here, you now see two bands, right? So the ribosome was chugging along, hits the slippery sequence, hits the triple helix, goes boop, one base back, and one base back. And then it's going to start over on the next reading frame. Absolutely batshit crazy. Good. Okay, so at the 5' end of this viral RNA, we have this massive polyprotein that's made, right? And that encodes two proteins through this crazy ribosome frameshift, right? Guess what? These two proteins are more than two genes, right? We've gone from one mRNA molecule already to two proteins. That breaks the typical rule of the cell. And then typically when a cell makes a protein, it just makes that protein, right? It can be processed a little bit. But here we have a bunch of different genes, like all of these different genes within these two large polyproteins. And within these two large polyproteins, we have proteases. Protease in biology, again, means cut. Protease means, it's a protein that cuts other proteins. So these proteases actually cleave at all of these arrows, these two polyproteins, down into a ton, right? A bunch of smaller components. Okay, so we've gone from one RNA to two large proteins, cleave, right? And we're down now into a bunch of small proteins that can actually finally do their function in the cell. Right? So we've gotten to a bunch of steps to now where the virus can say, oh, I can actually do my function. Okay, and these proteins are called NSPs for non-structural proteins. Makes sense, right? The idea here being that the structural proteins in a virus are those proteins that we need late for the virus to get back out of the cell. And the non-structural proteins are anything that's not that, right? So the proteins that are needed to do the molecular machinery and all of those things, those are non-structural proteins. All right, here's a structure of the viral protease from coronaviruses, right? So we already know what the structures of these proteases look like. And guess what? Proteases from viruses look unlike proteases from cells. And that makes them a good drug target. So a drug is a small molecule that binds to and blocks a protein from functioning. If you block a viral protein from functioning and the virus needs that protein, guess what? The virus can't function. But you have to find a thing that the cell doesn't do, right? If you block something that the cell also does, that's going to be very toxic for patients. So what we're looking for when we're trying to find drugs against viruses are drugs that will block the viral function without blocking the function of the cell. And proteases are a prime target. Some of the drugs that changed the game for HIV blocked the HIV protease, right? Again, the HIV protein that comes in and snips up a polyprotein into its components in order that they can, so that they can function, right? So there have already been studies that are looking for, novel therapeutics against coronavirus proteases, and they're testing in clinical trials as we speak, right? They're testing in clinical trials therapeutics that are known to block proteases from other viruses, including HIV. All right, what are the things that the virus has to make now? It has to make the machinery to copy itself, right? So the RNA-dependent RNA polymerase, also a drug target, by the way, right? Remdesivir is an RNA-dependent RNA polymerase inhibitor. It blocks the virus from copying itself and from making its genes. That is this machinery. Again, a bunch of NSP proteins you see on this slide, right? This is the viral polymerase. Some people call it the RDRP, RNA-dependent RNA polymerase. And so again, this is a multi-protein complex. There are a bunch of these NSP proteins that all interact with one another that make the core RNA polymerase enzyme, right? We have structures of the coronavirus RNA-dependent RNA polymerase. That helps us find drugs that may work against it, right? Again, test question for molecular biology students. What is the charge of DNA? What charge does DNA have? Pause your video or shout it out. DNA is negatively charged. So if you look at the charge of amino acids around the bit where DNA goes, right here, or sorry, RNA, this is an RNA-dependent RNA polymerase. RNA still has full negative charge, right? The residues around the RNA binding channel, where the active side of the enzyme is, largely positive to buffer and accommodate that negative charge. If you look at the conservation of amino acids, right? So, the evolutionary theory says if something is important, it's likely to not evolve, right? It's likely to be needed and therefore be there, right? So if you look at the conservation around the active site and the RNA binding channel is higher, most conserved. Those are important residues, right? So what you want to do is find a drug that binds in there to those most conserved residues, right? You can't mutate away from it, but that won't interact with cellular RNA polymerases or cellular DNA polymerases or any cellular enzyme. So we're hoping that remdesivir, an RNA-dependent RNA polymerase, blocks this thing, right? Stop it, block it, bleh, right? We kill this enzyme, the virus cannot copy itself. This RNA-dependent RNA polymerase does a crazy thing. Again, how many crazy things are we into this virus? We're into this virus by like 17 million crazy things already, right? This RNA virus, typically an RNA virus will have an RNA polymerase that makes its RNA, and it won't care too much about mutations, right? It says, I'm sloppy, I'm a virus, not every one of my offspring has to be functional, I can make mutations, it's not a big deal, right? This RNA-dependent RNA polymerase has what we call an exonuclease domain. That's an air checking domain. So the virus will say, oh, if I make a mistake, I'm gonna actually go back and fix that mistake before I keep going, right? What that means is this virus evolves very, very slowly, okay? And here's this is pretty colors, all of this nice stuff, right? This is a phylogeny. So it's sort of an evolutionary heritage, a map of the relatedness of these viruses, okay? And you can see that basically, long story short, things that are close together here are more closely related. Things that are far apart here, sort of up and down, right? Far apart are more distantly related, okay? And we can actually show this also in what we call a molecular clock. So if our ancestral virus is all the way back here on the x-axis, right? And the date is on the x-axis. So as we move, sort of, right? We're actually moving forward in time. And these are the full viral sequencing, the whole genome of the virus. How different is one virus from the other? Well, that experiment tells us. Across all of the viral genomes we've sequenced all over the world, right? How many mutations are there between them? And there are so few, y'all. There are so few, right? An average of around 10 total individual mutations. That's a single letter change in the whole genome. The most divergent virus we've ever sequenced, 20 bases. That's it, right? So this notion that there are 8 strains or 3 strains around the world, bogus, not true. We have no evidence for multiple strains evolving. That would mean sort of strains that are more severe and strains that are less severe, right? We only see a total of max like 20 mutations. This virus on average will only mutate around 25 bases a year. For an RNA virus, that is incredibly slow. HIV, flu, all evolve, all mutate, right? Mutations, of course, the raw material for evolution, right? Most viruses mutate much faster. RNA viruses mutate much faster than this. Okay. Now, we've gotten in the cell. We've made our polyprotein. We've made 2 polyproteins, right, for a ribosomal frameshift. We then cut those up into all their functional parts. We made an RNA-dependent RNA polymerase, right? And that is actually going to start encoding other RNAs, right, from the genome. So the other genes can now start to be made now that I've made my machinery to make those genes, right? So now the virus is actually doing work. Okay, and here's some of the other genes, the sort of structural genes later on that have to be made. And, again, viruses do crazy things, right? Every RNA that's made by this virus will have the same sequence at its 5' end that's encoded in the very far 5' end of the virus. That's called a leader. So the RNA polymerase binds there, right? It starts transcribing, it makes around 60 nucleotides, then it somehow dissociates and uses that 60 nucleotides as a primer to initiate synthesis elsewhere on the genome. We have no idea how that works, right? There's still a lot that's unknown. That is not how transcription works in cells or frankly in many other viruses, right? So every viral RNA that's made has a 60 nucleotide leader on its 5' end and a cap on its 5' end and a poly A tail, right? Makes sense. It has all of the things that RNAs need in the cell to actually be made into protein. Okay, so where are we at? We're talking about this life cycle, getting in and out of the cell, right? So we've gotten into the cell, we've made our replicases, our NSPs, right? And then we have to make all of these other viral RNAs that can then be made into proteins, right? Again, we need a cap, we need a tail. So researchers have been studying, right, the cell's machinery for capping. The cell's machinery for tailing RNAs is in the nucleus. The virus has to do that itself. And so what do we know and what don't we know? Well, we know that the virus does have poly A tails on its RNAs. Exactly how that occurs is still kind of a mystery. Here's a set of experiments that researchers did where they went looking for poly A tails on the 3' ends of mRNAs. And they did this in a way where they actually took the special moiety that's at the 5' end of mRNAs, which is a triphosphate. So a nucleotide with 3 phosphates on its 5' ends. That only occurs on the extreme 5' end of RNAs, right? It's normally capped. They remove the cap, and then they use that to ligate to the 3' end of another RNA. Now they know what sequences are around this ligation product, and they can use PCR to amplify the region in between. And they ask, is there a poly A tail there? And if there is, what is its length? And is it the same over viral infection, or is it different? So they run that down a gel, and they show that the length changes over time. They don't know what that means. They don't know how it works. That's sort of the extent of what we know, right? They did Sanger sequencing. Is it a poly A? They asked themselves, and they did Sanger sequencing to find out. And indeed, when you do Sanger sequencing, past this 3' UTR, you see A, right? Clear evidence that there is a poly A tail there. Again, mRNAs have to be capped. The viruses encodes machinery to add that 5' cap onto the 5' end of all of their RNAs, right? So, different viruses do this in different ways. Lots of viruses have capping machinery, and this requires a certain set of modifications to the 5' end of the RNA. The cells will do it in one way, the virus will do it in another, in a different location. Cell in the nucleus, virus in the cytoplasm, right? This goes all the way back to some chemists trying to identify from coronaviruses, right, these different chemical activities. And this is just showing you that the virus, the chemists separate out using chromatography the viral enzymes and look for particular activities in an in vitro assay and sort of map those activities to different proteins. We, of course, then go ahead and study those proteins well, solve crystal structures, right? And we see indeed that the viral capping machinery has a particular structure, binds to RNA in a particular way. And students, you should be asking yourself, does it do this in the same way as the cell? Because if it doesn't do this in the same way as the cell, it's potentially a good drug target, okay? And the answer is no, it doesn't quite do this in the same way as the cell. If we look at the enzymes, right, for viral capping machineries, we see that viruses do this in very different ways than humans do. So the human enzyme is down here. Let me circle it. Pen. Right? The human enzyme is here, and it looks different from like a vaccinia virus enzyme. Right? So we do think that these capping enzymes could potentially be drug targets. All right. So I want to end, we talked about virus sort of getting in the cell, copying itself, making different RNA products, right? Capping those RNA products, poly A tailing those RNA products so they can get translated by the cell's machinery, right? And that will allow it to make all the proteins it needs to get back out of the cell. But I want to talk about another crazy thing viruses do, in part because, I spent my PhD working on how a different class of virus, how phage, so viruses that infect bacteria, how they actually do the same process. Viruses are wily, right? And they don't just go ahead and go around and copy themselves, right? They actually go around and take stuff from the cell. They say, hey, I don't want the cell to be transcribing RNA. I don't want the cell to be translating protein because I want all those ribosomes. I'm the virus. I have to get in. I have to get out. I have to do my thing, right? It's sort of like that selfish friend you have or whatever. Insert student laughter here. That, is always taking, is always asking help for homework, but never offers anything, right? It wants to get its stuff done, right? And so the virus is actually strategically trying to, if it tries to do anything, is trying to tamp down what the cell is doing in order to co-opt those resources, okay? So it's been known for some time that, the virus somehow turns off cell gene expression. So if you look at the proteins that are being made in the cell that are non-viral proteins, the virus says, nope, cell, you have to stop making your protein so I can make mine, right? It's ribosomes, the cell's ribosome. Those ribosomes belong to the cell, y'all. Those are the cell's ribosomes. And here is the virus selfishly taking all of them. Rude, okay? The question is how, right? So if we look at this experiment, this is a protein gel, and you can tell that because of kilodaltons, right? So we're just looking at the protein output from the cell. And if we take this protein, NSP1, so non-structural protein 1, right? NSP1, again, one of those early proteins that's going to be made by the virus. And if we look at the protein output of the cell in the presence or absence of NSP1, you see that the wild type NSP1 actually leads to less protein being made by the cell. What the fuck, right? And it's tricky because the virus wants to stop this cell from making protein, but it can't block its own self, right? It can't just stop translation altogether. It just can't degrade all RNA because its RNA needs to get made, right? So the virus has a tricky thing it has to do, and it will have evolved mechanisms, right? The virus doesn't want anything. It will evolve ways in which to stop the cell from doing something and to activate its own gene expression. The question is how. Here we actually have a fair amount of detail of how this protein NSP1 is able to selectively degrade RNAs that are made by the cell and leave viral RNAs untouched, chilling, making itself, churning out. Okay? All right. So this is, again, just thinking through what does the virus want, right? The virus wants to make a ton of its proteins and to stop the cell from making any. It wants all the NTPs. It wants to make more RNA. I want those NTPs. I want all those ribosomes, selfish ass. Virus is the selfish ass, right? So the question is, what can stop RNAs from being degraded? What might be signaling on the virus, right, that, don't degrade me. Don't degrade me, don't degrade me, degrade the cell, right? And it can be that there are things like our particular RNA sequences. And again, RNA sequences can make structure. So in this experiment, they take two different versions of luciferase. So just two different genes that I can trace with their sequence, right? And it's going to do a blot, a northern blot for all you molecular biology nerds, right, against these two different sequences. So you have a full-length RNA that's capped and has a poly A and has this structured RNA called an IRES, an internal ribosome binding site, basically. It doesn't matter what it does. It's not actually even from coronaviruses, right? It's just a marker of an RNA that's particularly structured, right? And they're basically asking, does a structured RNA stop NSP1 from degrading that RNA? So could it be an RNA sequence or structure within the coronavirus genes that actually stops the NSP1 from degrading the virus's stuff, right? The virus says, I want to keep me. So if you look at this, if I add NSP1, any RNA, let me get my pen back out. I'm so good at teaching, right? If I have this IRES, so the full-length RNA, RNA1 have this IRES, but RNA2 does not, right? So NSP1, it gets rid of RNA2. If I don't have this IRES, that RNA goes away, right? And if I have this IRES in my thing and I add that full-length RNA, the NSP1 comes in and chews up to that IRES, but then it stops. That IRES stops it from moving any further, right? And you see that here. So the full-length RNA goes away, but it gets stopped at that RNA1, right, where their IRES is at. Okay? It doesn't get degraded past that, right? So the IRES is sort of a break. It says, nope, stop. Okay. Now, what could it be, y'all? What could it be? What could it be signaling? What isn't present in all of these mRNAs that could lead to a signal for NSP1 not to degrade them? Well, I have a poly A tail, but it's probably not that because the cell's RNAs have poly A tails as well, right? Mm-hmm. Yeah, uh-huh. It could be the this 5' UTR, this 5' leader, this thing that's present on all of the viral RNAs. Could that be protecting these RNAs from degradation? So, again, in science, we like to ask the questions, right? So we do this experiment where, again, so we just have a gene that we can follow. A lot of these viral studies are a little old, so instead of using the gene that we would use now, which is GFP as a marker, they use luciferase, same diff, right? I can follow the expression of luciferase in a very simple assay, all right? And they have, this RNA that is it has a promoter, it has a poly A tail, and it has luciferase in the middle. And they ask whether NSP1 can degrade this RNA, right? So if they're just looking at luciferase, is there luciferase? NSP1 didn't degrade. Is there not luciferase? NSP1 degraded. Okay. If I add wild type NSP1, so the functional protein, it degrades that luciferase really well. Right? But if I add the 5' UTR of the coronaviruses, so this bit of RNA that's at the 5' end, doesn't degrade at all anymore. Right? And that's true if I add the 5' and the 3' UTR, but if I add the 5' UTR alone, again, doesn't degrade. But if I add just the 3' UTR, it degrades just fine. So there's something in the 5' UTR, that 5' leader sequence, that signals to NSP1, hey, I'm a viral RNA. Leave me alone, right? So, these are the types of crazy cool molecular mechanisms that viruses will evolve in order to sort of decrease the expression of the host cell and co-opt the resources. And y'all, that was the last example that I wanted to show you. Again, I'm not talking too much about the structural proteins that help the virus get back out, but I just wanted to go through some of the molecular mechanisms that the virus uses to sort of go through its replication cycle in the cell. This has been Professor Joe Osmundson from my home here in Manhattan's Chinatown. Thanks to Dave for cutting this. Thanks to my roommate for putting up with me making a fake ass studio set in the living room for an hour. It's lunchtime, and so I think I need to let us all eat. Stay well. Don't die. Be well. Please be well.